Hey everyone, my name is David Mikhael. I'm a second-year Bioengineering Ph.D. student in the Berkeley and UCSF joint program. I'm part of the Neuromodulation Imaging Lab led by Dr. Melanie Morrison, and I'm co-supervised in this project with Dr. Andrew Moses-Lee, who is a psychiatrist at UCSF. And today I'm going to be presenting my project titled Treating OCD with Deep Brain Stimulation, Network Mechanisms. So basically what you will be seeing today is a neuroscience study on OCD, obsessive compulsive disorder, which I will introduce. But hopefully you'll have fun just listening to what we did and why we did it, and hopefully you see our research enjoyable. So I want to first start by introducing obsessive compulsive disorder, or OCD. 2 to 3% of people in the world meet the criteria for OCD at some point in their lives. Some of them are diagnosed, some of them are not diagnosed, and scientists think that the number of people who are not diagnosed but they have the symptoms are way, way more. OCD is actually one of the top 10 causes of illness-related disability in young people worldwide. And this is because it's chronic, and it's time-consuming to treat it, and it causes a lot of, I wouldn't say dysfunction, but it makes it hard for the person to work, function, daily life as comfortably as one ideally should be. Even with treatment, 30 to 40% of patients remain significantly symptomatic, which is what motivates approaches like neuromodulation and deep brain stimulation, which I will introduce. Don't worry if you don't know what this means yet. Before I go to the science of OCD, I just want to briefly mention a sentence or two about OCD for people who are not very familiar. With OCD, basically your brain is stuck in a loop of thoughts or ideas or compulsions, which is where the compulsive word comes in, and obsessions. By obsessions, we refer to things that we think about and we can't stop thinking about. On the other hand, we shouldn't be thinking about them, or we're basically excessively obsessing about them. By compulsions, it's actions that our brain tells us to take in order to satisfy these obsessions, which doesn't happen, which is why it's a loop. An obsession followed by a compulsion, and so on. It can be cleaning, it can be organization, or a wide variety of things. Briefly about the science of OCD. First, OCD, it's a circuit disorder. It's not a personality disorder per se. It involves abnormal brain activity in what we call a cortico-striato-thalamocortical loop. Each of these subwords is a brain area. And the dysfunction is especially in the orbitofrontal cortex, right here, the anterior cingulate, right here, and the striatum, which is a deep brain structure, also called basal ganglia. What happens is basically, as I mentioned, it's an overactive error detector. An overactive that's also not very rational most of the time. Regions like the anterior cingulate and insula, which is also like right here, it's a deep region, they fire excessively to signal that something is wrong. Either way, like I said, cleaning, organization, even when things are safe or complete. The person gets stuck in these habit loops, and these networks that should help us to shift from one action to another, they become rigid and not responsive to any attempt to fix them. Okay, this is why we are using deep brain stimulation to help treat this disorder. So, DBS is like a pacemaker for the brain. There are thin electrodes that are implanted by a neurosurgeon and an open brain surgery. It's an invasive surgery. In specific deep brain regions, and it's connected to a pulse generator in the chest. What happens is that the system delivers tiny electrical pulses that aim to modulate the abnormal circuits that I mentioned in the last slide, rather than destroying tissue. Because in the past century, a lot of these kind of techniques, they used very high frequency and high intensity electrical pulses to damage or to destroy an abnormal part of the brain to just remove its maladaptive effect, right? But now, instead of just lesioning a whole brain area to fix the problem, which usually even introduces more problems. We are just fixing it without damaging it, right? Imagine, yeah, like you are an electrician, you have a lamp that is not working. Instead of just replacing it, you fix it, so it works. It's also adjustable and reversible. So the settings of the stimulation can be tuned over time, either the pulse width, the amplitude. The pulse width is the duration of the pulse, or the amplitude, or the frequency. The system can also be turned off or even removed. It can be turned off using just a remote controller that's usually with the patient when they are sleeping, for example. And it's clinically used for movement disorders like Parkinson's disease, which is a movement disorder described by abnormal gait, some rigidity, tremors, a lot of other symptoms, and essential tremor, and is now being explored for treatment-resistant OCD, which is what we're doing here, and other psychiatric conditions. So the magic here is the beneficial effects start to happen once the device is on. We don't have to wait like months or weeks for it to show. Of course, over time, the brain circuit can adapt more and becomes more organized, but the speed is where actually the magic comes in. Very briefly about DBS history. You can also look this up online if you're interested, but in the 1950s and 60s, as I mentioned, it was lesion era stereotactic surgery. We were just lesion a brain area, either to see what happens when this area is inactive or to help, for example, remove seizures. 1987, reversible high-frequency stimulation, which paved the way for the current DBS. First FDA approval for a DBS clinical trial was for essential tremor on the VIM, which is an area in the thalamus deep in the brain. Early 20s, Parkinson's disease indications. People started to look for Parkinson's to be treated. Also, afterwards, dystonia, which is a movement disorder, and OCD, people started exploring potential DBS treatment for these conditions. And then around 15 years ago, people started using sensing and closed-loop DBS, which basically is a more adaptive type of DBS that gets feedback from the brain circuits it is trying to fix in order to reiterate on the parameter selection and optimize the set of parameters that bring the best clinical benefit using this closed-loop technique. Finally, adaptive and connectomic guided DBS, which basically is what we are trying to do now. We're trying to use what we know about the brain connectome or connections to guide where we place the DBS electrodes and make it more effective. Current DBS applications, as I mentioned, movement disorders, psychiatric disorders like OCD, epilepsy, and even for chronic pain. Current challenges with the clinical use of DBS, which is what motivates our study. First, we have a huge parameter space. We can tune and tweak a lot of parameters, and the parameter space is very wide. We have multiple contacts in each electrode, so each electrode has 4 or 5 contacts, and each of these contacts touches a different brain area. So which brain area in a millimeter resolution to stimulate is actually a tweakable parameter. We have a lot of other settings that we can tweak with no clear guidance. Trial and error programming. Usually, clinicians have the patients visit the clinic and they adjust the settings over time, relying on the symptoms, see if this frequency range helps symptoms more than that frequency range or amplitude, which is slow and subjective and inconsistent. Many patients never reach the best settings, where there is a lot of wasted potential, which is why there is less than 60% response rate in these OCD trials, despite it being an invasive surgery. Finally, which is where our part comes in, starting from the next slide, the lack of network biomarkers. Current practices do not use much post-surgery imaging, which is basically functional MRI or MRI while the DBS system is implanted, to see what it does to the brain and to understand which brain states correspond to good outcomes. Okay, this is where our work comes in. We are a brain imaging lab, so we run functional MRI and diffusion MRI and structural MRI, but for this project, it's mainly functional MRI based. Functional MRI is like a movie of brain activity. It basically tracks changes in the concentration of blood oxygen as a proxy for neural activity. When a neuron is active, it just employs a lot of oxygen to fill it with energy in order to do the activity and spike. Has great spatial detail, few millimeters resolution. It's also non-invasive. The patient just lies in the scanner and then go like 10, 15 minutes. It doesn't have risks associated, no harmful radiations or something. It's a decent temporal resolution mechanism, but it's not great. So it captures brain activity changes every 1 or 2 seconds, which is slow compared to things like EEG, but it still can provide us with good enough data. And we don't only study where the brain activates, but also how regions talk to each other. So is this activation and that activation synchronous? Do they correspond to the same function, or are they completely independent? And this is actually where we're focusing in the most here. So this is where also the word connectomics come from. Let's say, for example, near the frontal areas here, there is a lot of higher order cognitive processing. When you're solving math, you will see a lot of activations here, for example, that actually are synchronous with each other, that are correlated with each other, showing that they drive or they mediate the same function. And we call them functionally connected. Okay, brain functional networks. The brain is a network. It's not just isolated spots. Everything talks to each other. Regions or nodes interact through connections between them to form coordinated systems like the default mode, salience, and control networks. And don't worry, we will introduce these later. It's not just how strong connections are, but how they combine and talk to each other at a given moment. So at a given instant in the time series in the scan, certain regions can co-activate together, as I mentioned, forming a temporary state of the network. It can be like hyperactive, hypoactive, it can be a motor-related state, depending on what the person is doing. And these states flip over time, again, depending on what the brain is engaged in. And in OCD, some of these states may be overused or sticky, which is what we want to find. So parent error linked to threat monitoring and rumination can dominate, while more adaptive configuration appear less often. So that's the hypothesis, but we need more. Okay, our study. We have 41 functional MRI scans of 8 OCD patients under DBS. And each of these scans is conducted under a different DBS setting. Some of those settings were helpful in elevating the OCD symptoms, around 60%, while 40% were actually ineffective, so that patient didn't witness any improvement under these settings. And we have 2 patients out of the 8 who never responded to DBS under any of the configurations. Okay, as I mentioned, 60% improved brain, 40% hyperactive OCD-like brain, so no much benefit from G-brain stimulation. So the study goals is basically why? Find brain networks that characterize a therapeutic versus a non-therapeutic response to DBS for OCD. And the goal is, again, move from where the problem is to which network configuration supports recovery. Significance of the project. I think it's significant. I might be biased, but I don't think I am. You will see yourself. First, it's faster than trial and error DBS, as I mentioned. A lot of trial and error, a lot of parameter optimization, because it links stimulation to real-time brain network changes after surgery. So you can see after you've done the surgery, what brain networks now are tweaked to which direction, which areas are more connected or less connected with each other. And you can map these changes with what you know about what they do when it comes to symptoms. So in other words, you can map biology to behavior much faster. It shows how GBS helps, as I mentioned. And it helps us move towards brain-based biomarkers of therapeutic response. So we know that network patterns that track with symptom changes that could guide GBS settings. And it's a step towards personalized and adaptive GBS by knowing each patient helpful versus unhelpful brain configurations or states. Okay, an overview of our methods. We have, again, the brain scan while GBS is on. And then we ran what we call a co-activation pattern analysis. So these are 2 brain ROIs, just an example, a simplified example, 2 time series. So this is the functional MRI activity, again, blood oxygen level over time. And what we're trying to find here is common reoccurring patterns. So the green dashed lines here, the green dashed lines here correspond to time points where there is a positive activation in ROI 1, negative activation in ROI 2. So that's what we call a cap 1, cap again for a pattern, or brain state number 1. Blue corresponding for negative activation in the 2 ROIs, 3 positive and 2 ROIs. So basically it's a recurring spatial temporal pattern that is clustered together and then called the brain state. Again, these are just very simplified examples. Usually it's way more complicated than this, which is why we use clustering algorithms like k-means, if people are familiar, or Gaussian mixture model. I will not go into details in this. You'll hopefully see that paper and you can read more about the details. But what we end up with is something like this, where every color is a brain state that's basically driving brain's activity at a given point in time. And by I mean, when I say a point in time, I mean a frame or a time point in the functional MRI signal. So every 2 seconds, what is the brain doing under GBS? So I'm going to give an overview of 2 of the OCD relevant networks that would be helpful for you to track and understand our results. First is the default mode network. It's basically our default mode, like the name says. It supports self-referential, ruminative thinking and mind wandering. When you are like distracted, when you want to study your work, but your brain is just thinking about random things. It can become overactive in OCD, and you can get stuck in intrusive thoughts. Second is the salience or interoception network. And by interoception, we mean when you sense or pay attention to what your body is doing or your current state of body. If some of you do yoga or meditation, you're basically practicing interoception. And here we are saying that, or our hypothesis is that it's a helpful network state. It detects important internal and external signals and shifts our attention accordingly, which supports healthy engagement of our body awareness and adaptive action monitoring. So we are paying attention to what our body is doing internally and externally in a way that's not ruminative and that's not stuck in our thought loop. So that's our hypothesis. That's what the literature is proposing. But we now want to look at this in a data-driven approach and see if it actually holds. Okay, our main findings. So we actually found pretty much what we were expecting, but also with some additive details that help us have the complete picture. So on the left side here, you can see that the default mode network, these are the regions that correspond to the default mode network. And the brain visits these states less often during the functional MRI scan when the DBS configuration is therapeutic. So another way to say it, when a DBS configuration is effective, when the patient feels symptomatic improvements, the brain spends less time in this default mode ruminative bad state, right? On the other hand, the brain spends more time in the salience or action monitoring state. And when I say spend more time, just a very quick going back thing. Yes, here. So when I say spends less time, it's basically fewer frames show the pattern that corresponds to the default mode network, or more frames show the pattern that corresponds to the salience interoceptive network. And this characterizes the therapeutic response to OCD. These are our just two of the main plots. So you can see this is the salience state, this is the default mode network state. You can see that under therapeutic configurations of OCD, the brain spends fewer frames or less time in this default mode state, what we call fractional occupancy, which is basically how much the brain occupies one of these states. So default mode network is less represented or less occupied compared to the salience, which is significantly more represented under therapeutic configurations, which again is what characterizes a therapeutic response to DBS for OCD. Also, brain is more likely to transition into the salient state and less likely to transition into the default mode network state. So hopefully at this point you have the complete picture of what we mean by a state and by a transition, and how the DMN or the default mode network is different from the salience, and how they contribute to DBS in positive patterns. Okay, conclusions. Again, we are working towards optimized and network-guided DBS. Post-DBS fMRI shows that therapeutic DBS shifts the brain away from the default mode network or the rumination states and into salience or interoceptive states, which are linked to healthy monitoring of the body and external space. And these network signatures can serve as biomarkers to guide which contact to activate, which parameters to set, reduce trial and error programming, which takes up much time, and the effect is not necessarily optimal at all. We can end up in a local minimal, most of the time actually, and push OCD DBS response rates beyond the current 60%. So, final takeaway, take-home message. We integrate imaging-based network markers into future adaptive and closed-loop DBS systems for truly personalized neuromodulation. And one final word, if you're following the neurotechnology space right now, personalized neuromodulation is a big thing. A lot of the current companies, they invest millions and millions of dollars into things like DBS or even ultrasound stimulation, which is non-invasive, or transcranial magnetic stimulation, not just electricity, in order to, again, help fix the brain circuitry with the less invasiveness as possible and less time as possible. Thank you.
I actually have a question.
Yeah, sure.
So basically, like, if I'm understanding correctly, you're able to like characterize what brain states kind of correspond to like a therapeutic.
Mm-hmm.
Basically, like when it's working
Yes.
and when it's not working.
Exactly.
But you didn't like. Did you go into like what actual, like what the settings of the device were that corresponded to that, or do you know what I mean? Because I'm just trying to like.
Yeah.
I mean, like the ideal situation would be like, okay, you can basically, like you said, tune the parameters to basically ensure that a therapeutic response happens.
So at this point, no, and it's super hard because of the patient variability.
Yeah.
But what this helps is, what happens now is the neurosurgeon implants the electrode, the neurologist or the psychiatrist sets the parameters, then they let the patient go home. They wait for a few days to have the patient back and see if their symptoms improved or not. If they didn't, they blindly tweak, oh, okay, let's increase the frequency. Let's blah, blah, blah. And then they wait. But now, they can have the patient under an MRI, and they can see if the parameter that they decided to set today, this morning, produced patterns that we know are characteristic of a good response. So we expect, or we have a more probability of the patient actually coming back next week with good results.
Right. Yeah, it makes sense. So
Yeah.
it's like a valuable tool for them to have.
Exactly, yes, yes.